Well, thank you, Catherine, for the introduction. And the name of my talk today is Haploid Selection in Predominantly Diploid Scott Spine. So let's start. From all the diploid level systems that exist, the haploid diploid system is one of the most studied. And in this haploid diploid system, we have some organisms that spend most of their lives as haploids and only have a small stage of their life they spend as a diploids. Or we can have also organisms that spend most of their lives as diploids and only have a small stage of their life spent as a haploid. And why is important to study this kind of system? Like, what is important to study the haploid diploid system? Well, because we expect that having two different levels of ploidy within the same organism would pose different evolutionary pressures that could affect how the species evolve. And from the evolutionary point of view, we have different expectations of how selection should act in diploid versus haploid individuals. And our expectations are quite simple. We expect for selection to be more efficient in haploid individuals than in diploid individuals. And the reason for this is simple, because if we have a mutation in a diploid individual, either the mutation could be deleterious or beneficial. Meanwhile, the mutation is in recessive state, it will be able to hide from the selection in the heterozygous state. But if we have a mutation in the haploid state, this mutation is going to manifest immediately and it will reach fixation or it will be purged immediately. These expectations are known as the masking theory. And so, but this is theoretical expectations. And what about the empirical data to support this theory? Well, the empirical data for support this theory is kind of mixed. There is works that support the masking theory, and the majority of these works have come from experiments in experimental evolution, mostly done with yeast, and yeast is a unicellular organism. Another bunch of works that support the masking theory come from the studies of selection, the haploid selection over male, mostly male, if I'm not, maybe I'm incorrect, but I think most of the works are, or if not all the works are done in male gametes. But this is kind of a bit controversial because it's very difficult to separate the haploid selection per se from the sexual competition or the selection that you will have from sexual competition or pollen competition in the case of plants. And there are other works that do not support the masking hypothesis. And the reason for this is because we have another layer of confounding effects, and that is gene expression. Gene expression have their own evolutionary expectations by itself. We know that genes that are expressed in low levels are under more relaxed selection compared to genes that have higher levels of expression. We also know that genes that are widely expressed that have a wider breadth of expression, like for example, housekeeping genes, have a more constricted than genes that express in a narrowly manner, like say tissue-specific genes. So our aim with this work is to contribute to the understanding of the effects of, or to contribute to the understanding of the evolutionary dynamics that are going on in this diplohaploid system. And for that, we are using Pinus sylvestris as a model, Scott spine. And why is sylvestris a good model to study these kind of questions? Well, first, it is a tree, and trees have large population sizes, which makes selection efficient. Also, sylvestris has a haploid stage that is in the seed. The megagametophile tissue in the seed is haploid, and this is a multicellular tissue, and it has a life of approximately 2 years long. And this tissue is metabolically active and is also transcriptionally active until the germination time. And plus this tissue, the megagametophile tissue, is derived from the female gamete, which will allow us to avoid the confounding effects of sexual competition. So our hypothesis for this work is quite simple. if selection is more efficient in genes that are expressed in the haploid stage, we should be able to observe this as a decrease in the genetic diversity of those genes that are expressed in the haploid when compared to genes that are expressed in the diploid stage. So, and for that we have 4 expectations. Just to clarify you, our expectations are all related to the efficacy of purifying selection. So, in order to prove or to disprove our expectation, we took a 2-step approach. The first step of our approach was to generate RNA sequencing data from 5 different tissues in sylvestris. We generated data for the haploid tissue, the megagametophyte, and we generated data for 4 different diploid tissues in sylvestris. From this RNA sequencing, we were able to obtain expression values for all the genes that are expressed in these different tissues, and we were also able to obtain scores of tissue specificity for all these genes. And this score of tissue specificity ranks the genes from 0 to 1. 0 is assigned to genes that are widely expressed or genes that are expressed in the 5 tissues or in most of the tissues, and 1 is assigned to genes that are tissue specific, genes that have expression in only 1 out of the 5 tissues. So let's see. The second part of our approach was to generate genomic data from this. And for that, we use an exome capture where we put some of the genes that we identified in the previous state, and we use samples coming from one single population. So this is a brief resume of all the bioinformatic pipeline that we did, but at the end of all of this, we were able to obtain estimates of genetic diversity for our 0-fold or non-synonymous sites and for our 4-fold or synonymous sites. We also were able to obtain the distribution of fitness effects for our 0-fold sites. So let's jump to the results, to our preliminary results. Our first expectation was, if selection is more efficient in the haploid stage, so if selection is more efficient in the megagametophyte, or our haploid tissue, we should expect to observe a stronger negative relationship between the genetic diversity and the gene expression. And as you can see here, megagametophyte is not the tissue that has the most stronger negative relationship, neither for the 0-fold sites nor for the 4-fold sites. Besides that, even if we were to observe that trend that we were expecting, the dispersion in the data is so high that there is no difference between the different tissues. But I'm going to be very positive in here. For the relationship of the ratios non-synonymous to synonymous to the gene expression, we were able to get a glimpse of the trend that we were expecting. And I'm going to show you the third graph without the shading. And in here, we can see that the megagametophyte has the strongest negative relationship. I know that this is insignificant and the data is not different, or there is no different in reality in the tissues, but at least this is something in the trend of the thing that we were expecting to see. Another thing that is worth to note here is, I don't know if you can see my mouse. Another thing that is worth to see here is that the megagametophyte is the tissue that has the lowest level of expression of all the 5 tissues. So, expectation 1, there it goes. Our expectation number 2, if we were expecting to see lower levels of genetic diversity in the genes that are haploid tissue specific, and we have here 0-fold sites or non-synonymous, and we have 4-fold sites or synonymous, or what is also known as a neutral diversity. And as you can see in the graph, megagametophyte is not the tissue that has the lowest levels of diversity. So with reference to the non-synonymous to synonymous ratio, it's often assumed that you will get lower values of this ratio when there is selection acting on, or these low levels could be due to the action of purifying selection. And neither here in this relationship is the megagametophyte or the haploid tissue is not the tissue with the lowest values for this ratio. So our third expectation was that we should observe a skewness in the psi frequency spectrum towards red alleles. And this trend or this skewness should be more evident in the zero-fold sites. So, and the reason for that we were expecting this is because if selection is more efficient in the haploid stage, in the genes that are expressed in the haploid stage, we should not be able to see mid-frequencies mutation hanging in this area around the mid-frequency part, because the force of selection should push all of them to be riding along the low frequencies of the psi frequency spectrum. What we were expecting to see was more a kind of L-shaped psi frequency spectrum for the genes that are expressed in the megagametophyte. And that should be more evident in the zero-fold than in the four-fold, but we still should be able to see something in the four-fold. And we were not, we didn't got what we were expecting. So our fourth expectation is we should see a stronger signal of purifying selection in the genes that are haploid tissue-specific expressed. And what we are looking here is at the distribution of fitness effects of new mutations. And in the left part of the graph, we have mutations that are neutral or nearly neutral, and in the right side part of the graph, in this part here, I hope you are seeing my mouse, we have mutations that are strongly deleterious. These mutations, the way of these mutations, the effect of these mutations in the fitness is given or is given by the product of the effective population side by the strain of selection. What this means in practical terms is that having higher frequencies in the last category is an indicative of the efficacy of purifying selection. So what we have here is that we have megagametophyte, the haploid tissue, have a more efficient purifying selection. So this expectation, it was good. We were able to get this one right. So some conclusions. Okay, we were able to detect a strong signal of purifying selection in the megagametophyte tissue-specific genes, but the rest of our results are confounded with the level of expression. If you remember, megagametophyte is the tissue that has the lowest value of expression. So an expression level is a strong determinant of evolutionary dynamics. So our data is confounded. What is left for us now to do is to try to do a model where we could dissect the effect of the haploid selection from the effect of the expression level. So that's the next step. So this is my last slide. Many thanks to the people in the Puhayarvi lab, to former members, present members. And this work was done with the support of the Academy of Finland and the Biocenter Oulu paid for my salary. And so thank you a lot for listening. Questions, comments, suggestions are welcome.